We're going to cover cells. This is our way of getting back down from the great mountain of DNA transcription and translation. You need to stop doing that, Kat. Oh, shush. Hey, quit stabbing me. This is our way of getting down from transcription and translation and ending kind of where we began, looking at live organisms again. And we'll start by looking at cells in a more rigorous way than we did. We talked at the beginning of the class about the difference between prokaryotes and eukaryotes. Now we're in a position where we can look at that difference in a little more detail with a little bit more background. Okay, so if you can see the slide, there's two cells on it. The green speckly thing is a protist. Can everybody see all right? Everything okay? The green speckly thing is a protist, and then the blobby thing around it is another protist called amoeba. And inside the amoeba, there's this gooey looking stuff that flows through the cell. And this stuff, it's got lots of little grains and granules and bits and blibs and blobs in it. And it's called cytoplasm. Prokaryotes have this too. Every cell has cytoplasm on the inside, and it's a very complex solution. There's lots of water, there are salts, there are other atoms, there are proteins, there's other substances, and then of course there is the nucleus and various organelles that are floating in the cytoplasm or suspended in it. So cytoplasm is just the gooey stuff on the inside of a cell, and both prokaryotes and eukaryotes have this. And what surrounds the cytoplasm and what keeps it inside and keeps the outside outside is called the plasma membrane. This is a diagram of what it looks like on the level of molecules. Most of these names I would not expect you to know. You don't need to learn them for purposes of this course. What I want you to know is that the plasma membrane is made of a type of lipid. You might remember that lipids are one of the four classes of macromolecules that we talked about at the very beginning, right? Carbohydrates, lipids, proteins, and nucleic acids. Lipids also include fats and oils. And you have a layer of lipid molecules that naturally forms a very flat sheet-like or bubble-like structure when you embed it in water. And in blue, you can see there's lots of proteins that can be embedded in the membrane. You there? Okay, there are proteins and other molecules that stick into the membrane. They might cross the membrane completely. They might stick out of one side. They might stick out of the other. There's lots of things that these proteins do. On the right, there's a protein that looks kind of like a tube called a transport protein that allows certain molecules to pass in or out of the cell. It's kind of a gateway in or out. Some of these other proteins might enable the cell to respond to signals from the outside. Others of them might help cells stick together, or if this is an animal cell, stick to the extracellular matrix. There's lots of things that these surface proteins do. Don't worry about most of the names on the diagram for our purposes here. Just know that what you have is a layer of a type of lipid with proteins kind of embedded in that and sticking out of one side or the other, or sometimes both, of that membrane. And if this seems a little counterintuitive, the membrane isn't really solid. It's not like a rubber balloon or a latex glove or anything like that. It's technically a liquid, but it's a liquid that naturally tends to form a very thin film. And the best analogy that you would know is a soap bubble. If you've ever blown soap bubbles and maybe watched one up close, you might be able to see that soap is kind of, skittering around on the outside of the soap bubble. So that soap bubble is actually made of liquid, but the liquid forms an extremely thin sheet that balls up into a stable bubble, a stable structure that'll last for a while. That's the best model I can give you for what a cell membrane is really like. It's not like rubber. It's not a flexible solid. It's a thin but very stable film of what amounts to liquid. And those proteins that are in the membrane can actually move around within it, although they can't normally leave it. Hopefully I'm making a certain degree of sense. So we'll hit the next slide. Since those membranes are liquid films, it's easy for them to pinch apart or fuse together. If you've ever looked at soap suds, wow, that sounds kind of trippy. Hey man, you ever really looked at soap suds, dude? You stare at them for long enough and you take drugs. It's awesome. Actually, I'm kidding about the drugs. But if you look at soap suds, you'll be able to see small bubbles that will periodically come together and fuse into a bigger bubble. You could also, by poking them, I guess, make bigger bubbles and make them split into smaller ones. Bubbles can always come together and can always pinch apart. And it's the same for a cell membrane and also for the membranes inside a cell. Because they are these two-dimensional films of fluid, they can easily pinch apart and easily come together. The context where you've heard of this happening is in cell division. Remember that in cytokinesis, one cell will kind of pinch off into two. You couldn't really do that with a rubber balloon. You could kind of squeeze a rubber balloon down and then tear the rubber. But the minute you let it go, the two halves would immediately deflate. So it doesn't work so well for a solid membrane, but for a thin film of liquid, like soap bubbles or cells, the membrane can break and come back together, and little bubbles in the membrane can bud off or can join. You have this very dynamic system where little bubbles are always fusing or budding off. Okay, so stare at your soap suds the next time you do the dishes, and you'll have some idea of what cells get up to. So here's a slice through a prokaryote. It's a bacterium. Since it's shaped like a rod or a sausage, hopefully you remember that this is a type called a bacillus. This one has been sliced in two and examined using a gizmo called a transmission electron microscope. We don't have this type of microscope at UCA. They've got one at UALR. And if you slice this through, you can see there's a cell wall outside the cell membrane. Most prokaryotes have that, although there's a few that don't. Inside, it's all pretty much just a grainy mass. In the center, there's this kind of lighter gray-looking, stringy kind of plate of spaghetti noodles region. That's the DNA. And that central area where a prokaryote keeps its DNA is called the nucleoid. It's not the same as a nucleus, because a nucleus would have a membrane sac dividing it from the rest of the cell. In the case of prokaryotes, there's no sac. It's just a big blob of DNA in the middle That's the nucleoid. And some of those black specks that you see around it are going to be ribosomes, and those are just kind of floating free in the cytoplasm. Transcription and translation all happens in the same place. I think I compared a prokaryote to that big indoor practice facility that the football team has, which is basically one giant room covering half a football field. Same thing in a prokaryote. Transcription and translation are happening in one single room. A difference between prokaryotes and eukaryotes that I didn't mention is that prokaryote DNA is organized into loops. Eukaryotes have chromosomes, and a chromosome is basically one long strand of DNA, one long piece of string that's got a beginning and an end. Bacterial DNA forms these loops, and the DNA that makes up that nucleoid is one ginormous loop. In addition to that, a bacterium can have a number of smaller loops containing genes called plasmids. That's a picture, again, this is taken with a transmission electron microscope of a couple of bacterial plasmids. Bacteria can pick up plasmids from the environment. When bacteria have, bacteria can pick up plasmids from the environment, and also when bacteria have what passes for sex, they can pass plasmids from one to the other. One of the problems with antibiotic resistance in bacteria is that plasmids often contain genes for antibiotic resistance, and those plasmids can be copied and traded among bacteria very quickly. Bacteria have this DNA kind of file sharing system, like SoundCloud or something like that, which enables genes for resistance to spread among bacteria very quickly. When we talked about Griffith's transforming principle, that's ultimately what the bacteria were doing. They were picking up plasmids from the dead bacteria they were with. So bacterial DNA is loopy. Eukaryote DNA is linear. Each chromosome is a long piece of DNA with two ends. Hopefully you still remember that eukaryotes have a defined nucleus. Oh, I think I wrote nucleus here. That's a typo. It's a nucleus with an N as in November. In the picture that you're seeing, these are eukaryotes that have been stained using a technique called fluorescence microscopy, which we can do at UCA. If you take some of our upper division electives, we can teach you how to do this. And the stuff that's inside the cell is mostly stained red and green. But inside every cell, there is a great big blue blob. That's the nucleus. And the nucleus is surrounded by a double layer of membrane called the nuclear membrane. Which makes a certain amount of sense, I guess. That's a slice through, it happens to be a plant cell, but it counts as a eukaryote. This is also taken with this gizmo called the transmission electron microscope. The grainy stuff that you're seeing in the middle is the nucleus. You have some other organelles and things kind of out to the sides, but most of what that slide shows you is the plant cell nucleus. And inside it, there's a darker spot called the nucleolus. You can actually see this with the light microscope if you stain it with the right stains. The nucleolus happens to be the place where ribosomes themselves get made. Ribosomes are made in the nucleolus, and they pass from there out of the nucleus to the cytoplasm where they're needed through what we call nuclear pores. This is a eukaryotic cell that was specially treated by being frozen and then hit with a very narrow but tough and sharp blade. what that did was split the eukaryotic cell through the middle, and no, hello, cat. Yes, you can't get up here. I'm busy. Right. Yes, you're cute. Shut up. Okay, right. Okay. Yeah, this is a cell that was flash frozen and then broken very quickly and split. And the crack tended to follow the contours of whatever was in the cell. This is a process called freeze fracture. So what you're actually looking at is the surface of the nucleus, and you can see it's covered with little bumps. Each one of those bumps is a set of complex proteins called porins, and the porin proteins make up nuclear pores, which act like little trapdoors that can open and shut. So ribosomes can get out of the nucleus, and messenger RNA can also get out of the nucleus, and certain proteins can pass into the nucleus, and so on. Those nuclear pores are the way the nucleus communicates with the rest of the cell, and the porins just act like little gates that can swing open and then swing shut. Okay, you hanging in there so far? I'll take silence as consent. Okay. The cell's packed full of membranes inside the cell. Of course, membrane makes up the cell membrane on the outside, but there's also a lot of folded membrane inside a cell. And there's a network of folds and sacs and tubes of membrane inside the cell called the endoplasmic reticulum. Okay, endo means it's inside. Plasmic should make you think of cytoplasm. It's inside the cytoplasm. And reticulum is Latin for a net, a little net. So the little net inside the cytoplasm is the endoplasmic reticulum, and if you can't be bothered to remember that, say ER. The ER is a transport network within the cell. Many cells make proteins and export them out of the cell, and they go on to somewhere else. So they're made and they're exported via the ER, via the endoplasmic reticulum. They get packaged for export out of the cell in a structure called the Golgi body. Not that you asked, but Camillo Golgi, who discovered this thing, won a Nobel Prize back in 1905. He's famous in his field, I suppose. And if you imagine the cell as being like a factory, you can think of the ER as like the factory floor where things get made. And the Golgi body would be packaging and shipping, where the proteins that get made are sorted and prepared for getting sent out of the cell. And there's also a various number of small little membrane sacs in a cell. Small sacs are called vesicles. Larger sacs are called vacuoles. These might be used for storing substances or for transporting them. There's a lot of things that those can do. The whole thing is called the endomembrane system. And again, endo means inside. The endomembrane system is the system of membranes that's inside the cell. What you're looking at there is another cross-section through a cell taken with a transmission electron microscope. And what I trust you can see is a whole bunch of parallel lines. You're looking at a slice through the endoplasmic reticulum. What it consists of is a whole bunch of membranes folded on top of each other into this kind of network of tubes and pleats and sacs. If you wanted to make a model of the ER, you could just take a bed sheet and kind of fold it in on itself a couple of times. Imagine you're looking at a folded bed sheet from the side, you'd see the same kind of flat, edges of the sheet that you're seeing in this picture right here of a slice through a cell. This happens to come from a cell that makes lots of protein for export. If you want an example of a cell that exports lots of protein, one that is close at hand, you could, if you put your finger on the angle of your jaw, you can feel your salivary glands. Cells inside those make a protein. It's an enzyme that breaks down starch called amylase. And then once the cells have made it, they have to send it out of the cell so it can go into the saliva and then into your mouth and break down starchy foods that you eat. So this could be from a salivary gland cell. I'm not sure if it is or not. But if you look carefully, you'll notice that some of these folds of ER are speckled with lots of little dark specks. Those are ribosomes. The ribosomes translate messenger RNA into protein, and then the protein passes through all of these folds and passages in the ER. And ER that has lots of ribosomes on it, by the way, is called rough ER. So this is rough ER that you're looking at. And if there's rough ER, there's also smooth ER, and you're looking at a slice through some of that. This smooth ER looks kind of more like you've cut into a network of tubes. Smooth ER can be used for making things that the cell needs that aren't proteins. Fats or other lipids might get made in the smooth ER. Carbohydrates might get made on the smooth ER by enzymes that are there. Your liver does the work of detoxifying certain poisons that you might take in. We talked about alcohol as an example a while back. And liver cells would have lots of smooth ER because this is where they would actually break down potentially dangerous substances in the body. Okay, so whatever gets made on the ER will pass through all of those folds of the ER and end up in a structure that looks a little bit like you took 10 deflated beach balls and stacked them one on top of the other. It's a stack of flat sacks of membrane. You're looking at a slice through one. In 3 dimensions, it would look a little bit like a stack of pancakes, except the pancakes would be hollow. There would be space inside. And again, this is another diagram where you really don't need to learn the details in the diagram itself. Let's just say that anything that's secreted from the cell passes to the Golgi. And remember, we talked about how soap bubbles can constantly break off and rejoin. Well, bubbles of membrane from the Golgi are constantly budding off. They're called vesicles. And vesicles can bud off the Golgi, move to the outer membrane, and then fuse with the outer membrane like a small soap bubble joining a large soap bubble. And in the process, they release the contents to the outside. The process, by the way, if you really want to know more, stupid Greek names, is exocytosis. Cyto means the cell, exo means out of. Exocytosis is getting things out of the cell. And you get them out of the cell by budding these little bubbles off the Golgi, and they travel to the outer membrane of the cell and then fuse with it, and the contents can spill out. Okay, vesicles are just small, and they're often used for transport or export. You're looking at a slice through some plant cells, and what I want you to focus on here is lots of empty spaces Much of a typical plant cell is taken up by a much bigger fluid-filled space made out of membrane. It's not a vesicle. We call it a vacuole. And that empty space in the middle of a plant cell is a vacuole. The vacuole normally contains water. When a plant has lots of water available to it, the vacuoles fill up and they swell. And just like a water balloon, when a vacuole is full of water, it is rigid. So, for example, if you have got some very nicely watered lettuce or spinach, the leaf is fairly firm and fairly crispy because of the water pressure inside those vacuoles. But of course, if you leave your lettuce or your spinach outside and it starts drying up, the lettuce wilts and becomes very floppy because you've lost that internal water pressure in the vacuole because the cells are losing their water as the leaf dries out. So, yeah, even without microscopes, you can demonstrate in your own kitchen even what central vacuoles do. They provide structural support to parts of the plant that don't have xylem and phloem. Okay, moving right along. I'm not hearing anybody, so I'm assuming everybody is receiving and understanding this. Please interrupt if there's something you need me to go back to or explain. Okay, I'll assume that this is going great. Okay, here you can see more rough ER at the upper left, but most of this picture you've got this thing that looks kind of like a stripy sausage. This is an organelle that only eukaryotes have. Prokaryotes don't. And by the way, prokaryotes sometimes might have vacuoles and vesicles, but they don't have an ER or a Golgi. They also don't have this thing called a mitochondrion. Not that you wanted to know, but mitos is Greek for a thread. This is kind of a short and stubby mitochondrion, but they can be a lot longer and more thread-like. And apparently this was a meme going around for a while, that mitochondria are the power plants of the cells. I think that's going around as an example of the kind of useless crap you learn in college. But you absolutely depend on your mitochondria because most of the chemical energy, not all, but most, that you extract from your food is extracted in the mitochondria. This is where the work gets done of taking the food that you eat and converting it into a form that can power everything that a living organism does. Almost all of the energy that you need to, even, think, be conscious, listen, and of course move, ultimately comes from mitochondria inside your cells. And yeah, it's not a perfect metaphor, but you can think of them as the power plant. I'm going to skip the biochemical magic that explains how mitochondria do this. It's really cool, but we just haven't got time. And I'm just going to give you a taste of it. This is a diagram of a compound called adenosine triphosphate, otherwise known as ATP. You might notice that on the right, you have a double ring of containing some nitrogen atoms. That's actually the base adenine. You might remember that from nucleic acids. And then to the left of it, you have a little pentagon with some oxygen stuck to it. That's our old friend, the sugar ribose. So you've got a base and a ribose, and instead of just one phosphate group, you have three instead of one. Yeah, this is basically a nucleotide. You use the same kind of thing, the same kind of molecule for making your DNA and for carrying energy around the cell, which is kind of neat. So there you've got ATP, and on the left you can see you've got three Ps, three phosphorus atoms, each one of which has oxygen stuck to it. It's got three phosphate groups, whereas in DNA there would just be one. And what ATP can do is give up one of those phosphate groups. It's shaded here in pink. At the top you can see there's a phosphate group that is stuck to an ATP. Incidentally, you don't really need to know this, but ATP tends to form these weak bonds with magnesium atoms, but don't worry about that here. Down at the bottom, you can see that that phosphate group has gotten transferred onto another molecule. It happens to be the amino acid serine. So what you have now is the adenosine triphosphate becomes adenosine diphosphate, ADP, and that phosphate group that was on the ATP is now stuck onto a different molecule. The reason this matters is that that gives an energy boost to that molecule. Remember, chemical bonds store energy. So that serine not only has a phosphate stuck on it, it has some additional energy that might be used to break down or otherwise modify the serine. There's a lot of chemistry that doesn't happen unless you can stick a phosphate group on first. This means that ATP is sometimes called the currency of metabolism, or you could call it the fuel of metabolism. All those reactions that need energy input are fueled by breaking down ATP. A familiar example is your muscles consist of two proteins that have the shape kind of like cables running alongside each other. They're called actin and myosin. Part of the myosin protein will actually break up ATP. So ATP molecules will come up to the myosin protein, give up a phosphate, and giving up that phosphate causes the myosin protein to change shape. When the myosin protein changes shape, what it can then do is slide past the other protein, actin, and your myosin cables and your actin cables slide past each other, breaking up ATP into ADP and phosphate as they go. And that sliding action, where the filaments do this, that's how muscles contract. Every little twitch of a muscle burns, I don't know, hundreds of thousands of ATP molecules just to happen. Each one gives up a phosphate and transfers energy to the myosin protein. And that's ultimately how this sprinter is getting off the blocks. He's going through ATP. Sorry, I Googled the phrase feel the burn, looking for something that would express this next slide, and I ended up getting that. I assume the gentleman in this picture has definitely felt the burn a lot. And the burn is the fact that you burn an amount of ATP every day, breaking it down into ADP and phosphate. If you could burn all of it at once, it would be equal to your own body's weight. Now, it's not, because your body's not made of nothing but ATP. But what happens is that every ATP molecule in your body cycles 500 to 750 times every day. And by cycle, I mean it loses a phosphate in driving a chemical reaction like muscle contraction. And then it gains a phosphate by the chemistry that takes place in the mitochondria. So every molecule of ATP in the body gives up energy to become ADP and then picks up energy to come back to ATP and goes through that cycle something on the order of 500 to 750 times every day. You're constantly, always, always, always burning ATP by breaking off a phosphate and then regenerating it by sticking the phosphate back on. And what sticks the phosphate back on is chemical magic. Well, not really magic, but chemistry taking place inside the mitochondria. Like the cartoonist Larry Gonick once said, I can't tell whether I'm taking my mitochondria for a walk or vice versa. Your mitochondria might be taking you for a walk. And again, we don't have time to get into the stuff that the mitochondria are actually doing. If you're really curious, you can look up something called the tricarboxylic acid cycle and the electron transport chain. You can look that up on Wikipedia if you really want to know, but I'm not going to test you on it. But what you need to know is ATP. You could think of ATP as being kind of like the cash that you've got on hand. You might constantly be getting more from, an ATM or something, and then spending it and getting more and spending it. If you're not living paycheck to paycheck, you've got other forms of reserves as well. You might have a savings account or stocks or a retirement account or something like that. Those are a little bit harder to touch, but you can burn those for cash if you need to. You might also have valuable things that you can sell for cash if you need to. But what's kind of going through the economy the most on any given day would be cash and the stuff in your checking account. You can think of ATP as a little bit like that. You're constantly burning through that. You store energy in the form of carbohydrates and fats, and you convert those to ATP. And if you don't have enough carbohydrates, you can, of course, start breaking down fats. If you're starving, you might also break down the proteins in your muscle, and you can feed that into your mitochondria. So your mitochondria will take the breakdown products from breaking down fat, breaking down protein, breaking down almost anything, and ultimately can use it to generate ATP. So your body has different levels of energy reserves. Just like you probably have more than one type of money than just the bills and coins in your pocket, or whatever is in your checking account. You probably have other resources you can draw on. Right. Hopefully I've just dazzled everyone with my brilliance. Let's keep going. Way back in the second lab, you saw those green dots called chloroplasts. Chloroplasts are the organelles where photosynthesis takes place. And again, there's some beautiful chemistry here that for our purposes, we're not going to get into because we haven't got time. Let's just say that chloroplasts are able to harness solar energy to build larger molecules that the cells need out of small molecules, carbon dioxide and water. What chloroplasts do is actually kind of the reverse of what mitochondria do, where larger molecules get broken down to carbon dioxide, water, and ATP. In chloroplasts, energy in the form of solar energy comes in and assembles water molecules and carbon dioxide molecules into larger molecules. Photosynthesis is kind of like respiration, which is what mitochondria do, run backwards. And that's all I'm going to say about how that works. But plants, of course, use photosynthesis to build large molecules. And then when you eat a salad, you take those large molecules and break them down for the energy that you need to run your own metabolism. And so it goes. It's the circle of life. Right. Holding all of this together inside a cell is a network of filament-like proteins called the cytoskeleton. Cyto, again, means cell. So a cytoskeleton is just the cell skeleton. There are some types of protein that make up what are called microfilaments. Others may be short tubes or long tubes called microtubules. They support the cell. They enable it to keep its shape. In many cases, they enable the cell to move. What you're looking at there are some cells that have been stained with a fluorescent stain, and the nucleus is that yellow blob in the middle, and then the stuff that looks like cotton candy surrounding it, the stuff that looks like hair or fuzz, are the protein microtubules and microfilaments that make up the cytoskeleton. So that's what that looks like. And then the last thing I'm going to cover for today, and we'll pick this up on Thursday, is that some eukaryotic cells, although not all, have what look like little hairs sticking out. This is a view, I believe, of the inside of your lungs. And your lungs are lined with epithelial cells. You might remember that's one of the 4 basic tissue types. And these cells in particular have little hairs that beat back and forth. They can move on their own. Well, they need ATP to do it, never mind how, but they beat back and forth. Some protists will use these little things to swim through fluid. In your lungs, these things beat to push fluid past the cell. The fluid does the moving and the cell stays where it is. So your lungs are constantly making mucus, and the mucus gets pushed along by the lungs. And then ultimately, when you cough, out of the lungs, so it doesn't just stick around and clog things. If you have a very long hair-like structure like this, like a sperm cell, it's called a flagellum. Flagella is the plural. If you have lots of short ones, it's called cilia. The singular is a cilium. The plural is cilia. And they have the same basic structure. The only real difference is that flagella are relatively long and cilia are relatively short. Cilia, if you're curious, was Latin for your eyebrows. So cilia look a little bit like the hairs that make up your eyebrows. That's where the word comes from. If you know the word supercilious, that's Latin for eyebrows above, meaning you're walking around everywhere with your eyebrows lifted because you feel like you're superior. That's what supercilious means. Sorry, I can't do it really well. All right. Where it came from is what we're going to talk about on Thursday with the work of a woman by the name of Lynn Margulis, who worked out where all of this incredible eukaryotic complexity of structure actually comes from. But for right now, I'm going to stop sharing. And do we have any questions I can answer? Or even questions I can't answer and I'll make something up because I've got a PhD.
Hey.
Hey.
Can I get a question? Like, it's not the lecture, but I have a question.
Okay, let me stop recording.
Okay.
And then I'll take it. Does anybody have a question about the lecture material? Stop. All right. With that, I will stop recording. Stopping now.